Alright guys, so this is going to be our last lesson in the evolution unit. And it's going to be taking a look at evolution during the developmental processes and how that can possibly lead to some new traits. And so it's really kind of combining two topics. So we're going to look at development of an organism and then how the development stages could lead to change over time. So you hopefully watched or started watching What Darwin Never Knew, and pretty much what that's about is how some of these evolutionary changes can arise just due to simple mutations in maybe developmental genes and things like that. So one part of the essay was, pretty much define what is Evo Devo, or evolutionary developmental biology, and essentially it's looking at evolution through developmental processes. And so just as organisms have changed over time, so have some of these mechanisms that lead to the development of an embryo. They have also changed over time. And so there's four kind of overlying principles of Evo Devo, okay? And if I were you, I would kind of star this slide. This is definitely one of the main things you need to know about evolutionary development. And so we're going to kind of walk through them, and then we're going to look at some examples. So this particular lesson is kind of like gene regulation, and that it's very technical. It's very complicated. I don't know that it's fully understood. And so everything that we're looking at is kind of an overview of some of the things that could be involved in changes in developmental pathways. And so these are kind of just four main concepts. All right, so number one, many groups of your kind of complex multicellular organisms. Obviously, when we're talking about evolutionary development, we're going to be focusing on those organisms that are multicellular and they have many developmental stages. So we're primarily talking about plants and animals here, but they share similar molecular mechanisms. And so when it says molecular mechanisms, that just has to do with things like your DNA, your protein synthesis, some of those same kind of basic little tiny subcellular mechanisms, okay? So they share similar molecular mechanisms for morphogenesis. Now, morphogenesis is going to be the formation of like body parts, and pattern formation. So pattern formation would be like where the head is, where the tail is, where the arms go. In terms of plants, it would be, what is the stem? What is going to become the leaves? What is going to become the roots? So number two, the molecular pathways that determine these developmental processes are independent. Okay, so these molecular pathways operate independently of each other. So that would be modularity. So if we're talking about developmental processes, it could be that the mechanisms that lead to the production of limbs is going to be separate than the molecular pathway that leads to the formation of the nervous system. Okay, so each of these molecular pathways are independent of the other. So if you had a mutation or a change to the pathway that affected limbs, it would not necessarily affect the one that was operating to control the nervous system development. All right, number three, changes in the location and timing of expression are important in the evolution of new body forms and structures. And this is really what you're seeing in the video, What Darwin Never Knew. And that we know that Darwin had no access to DNA or genetic information. At the time of Darwin, they pretty much had access to fossils. They had access to homologies, but they couldn't look at DNA. It was, almost 100 years later before we knew the structure of DNA. So now we know that depending on where the genes are located or when they are expressed in this developmental process, those could potentially lead to new body structures, okay? And then number four, development produces morphology. Okay, and so once again, morphology has to do with the formation of body parts. And what this one is all about is that the evolution of these body parts are typically modifications of existing structures. Okay, once again, kind of like what you're seeing in the What Darwin Never Knew. At the end, I don't think you've gotten there yet, but at the end, you're going to talk about this fish that kind of use these fins almost like as limbs in the front. And so essentially, it's not that we're just coming up with brand new things out of the blue, but most of the changes in structures are usually modifications of things that already exist. Okay, so once again, These are the 4 main principles of Evo Devo, and I'm going to kind of tell you to star a couple of slides, and this is definitely one of those that's pretty important to the overall lesson. All right, so what is development? Well, development are those processes by which we go from a single cell all the way into a multicellular organism, and it develops from the zygote to the embryo all the way up to the full-grown baby or kind of offspring. And so what we're going to look at are some of these processes that occur in the embryo and from zygote on, okay? So there are 4 main processes of development. This is another slide that you need to star. Okay, so number 1 is determination. So remember that early on you start off as a zygote. That zygote undergoes rapid cell division. It forms a hollow ball of cells. And very early on you have cells that are not specific. Okay, so determination is essentially setting the fate of what that cell will become. So down the road, will that cell become a muscle cell? Will it become a skin cell? Will it become part of the liver? And so this is essentially the very early kind of determining what will that cell be. Number 2, differentiation. This is when that cell actually becomes that. Okay, this is when that cell becomes specialized and becomes differentiated to be a specific type of cell with a specific type of function. Number 3, morphogenesis. Okay, so morph normally refers to change. This is essentially going to be when those cells become body parts. So this is when we get like organs and organ systems, tissues. So you think about your organization of life, the cells form tissues, tissues form organs, organs form organ systems. And so that's essentially what's going to happen in morphogenesis. And then the final stage is going to be growth. We're going to see an increase in size. Cell division is going to occur, your cell cycle, mitosis, and then that embryo will get larger, okay? So number 1, determination. It determines what the cell will be. Differentiation, the cell becomes that. Morphogenesis, it forms into body parts, and growth, it gets bigger, okay? So once again, this is another slide that you need to star. All right, so here's kind of what it looks like for an animal. Once again, a single-celled zygote undergoes rapid cell division, forms a hollow ball called a blastula. Gastrulation forms like the basic digestive tube. Your embryonic germ layers, your endoderm, your ectoderm, your mesoderm. We'll take a look at this when we get to animals. And then you have like your early stages of your embryo and then your full-grown organism. All right, so the cell fate. Now, as we're going through the rest of the notes, we're going to look at some things that can affect these 4 processes of development. Okay, so when we're talking about cell fate, we are pretty much talking about that differentiation or the determination stage in your development. So what determines what that cell will become? A big part of it is going to be gene expression, which, as I mentioned before, is very technical and not necessarily fully understood. And a lot of that gene expression and the chemical mechanisms are going to be controlled by the extracellular environment. So pretty much those things that are on the outside of the cell. A lot of it is chemically based, okay? It's almost like we're combining cell communication notes with gene regulation notes, both of which were very technical, okay? So it's a combination of changes in gene expression, when genes get turned on, how long they get turned on, which genes are turned on or off, as well as some chemicals in the outside environment. So once again, determination is a commitment, okay? When it actually becomes that, that is when we have differentiation. So these cells that are not yet determined are known as stem cells, okay? And so they are undifferentiated, and we can classify stem cells into 3 main groups, okay? This is another slide that you need to star. We have kind of 3 vocabulary words: totipotent, multipotent, and pluripotent. And we're going to talk about them in terms of potential, okay? So totipotent have the most potential, okay? I like to remember that they can become totally any cell, including the whole organism. Now, pretty much the only cell that is totipotent is the zygote itself. So when sperm meets egg and you have that original diploid cell, that zygote has the potential to become any cell type, from muscular to nervous to skin to bone to blood, okay? But you could also take that zygote, and from that zygote, it can develop into the whole organism. So totipotent can pretty much form totally anything, and they are going to have the most potential. Now, I'm actually going to skip down to pluripotent next, because in terms of potential, pluripotent have the next highest potential. And they can pretty much form all cells of the body, okay? But if you were to pull one of these cells, they couldn't necessarily form the whole organism, okay? So they're kind of one step down from totipotent. So they could still form all different cell types, but you couldn't get the whole organism. And these are what, this is kind of the word that describes our embryonic stem cells. I don't know if you've ever heard that term before. A lot of times it's brought up in the news. But your embryonic stem cells have kind of been controversial because they potentially could become any cell type in the body. And so there's a lot of potential use for them in terms of medicine and cures. But you have the dilemma of if you take these cells from an embryo, it destroys the embryo. And so you kind of get into the process of where does life form. And so there has been some conflict over using these embryonic stem cells. Now, the third, I guess, in potential is going to be multipotent. Now, I know that this word can be a little misleading because it says multi, which means many, and they can become more than one type of cell, but it's typically only a few cell types. Okay, so these are going to be like the stem cells that you find in your bone marrow, what we call our hematopoietic stem cells. And these are the ones that can produce all of your blood cells, your red blood cells and all the various white blood cells. You have mesenchymal stem cells, and these come from the mesenchyme, and these can produce a lot of your different connective tissues like bone, blood. Mesenchyme could also ultimately lead to muscle. And so these mesenchymal stem cells come from the mesoderm, and they can only become a few types, okay? Now, these are the ones that we would probably most likely refer to as adult stem cells, and they're definitely more limited. So in terms of stem cells, totipotent have the most potential. They can become totally anything, including the whole organism. Pluripotent are next in potential. They can become pretty much any cell type, but they could not become the whole organism. And then multipotent can become a few cell types. So of the three, they have the least potential. All right, so here's an example. Your stem cells, once again, are undifferentiated cells that have the potential to become other cell specialized types. All right, so as I mentioned, these embryonic stem cells, these are your pluripotent, but they can kind of be induced. And so there was a lot of study, it's been several years ago now, and I'm sure they've come a long way since then, but they were really a hot topic. I would say probably about maybe 10 to 20 years ago. But essentially we can, with the right chemical signaling, they can be induced to form different types of cells. And so like I said, they had a lot of potential. They could potentially repair damaged tissues like muscle and nerve that won't naturally regenerate and fix themselves. But we do have the ethical issues that arise, okay? Now, several years ago, there was a process that was discovered where you could take something like skin cells, which were from an adult, and you could almost literally like turn back the clock, and you could cause them to become these induced pluripotent stem cells. And so you could take skin cells from an adult, turn back the clock, and with the right chemical signaling, take them back to an undifferentiated cell, and then turn them into whatever you wanted them to be. Now, the good news is that immune response would be avoided because it came from that person, but also you're not using the embryonic stem cells, which are controversial. So I would imagine in a lot of our studies today, they're using these induced pluripotent stem cells. All right, now gene expression. Once again, this is very technical. Gene expression is essentially determining which genes are getting expressed and which genes are not. And we know that essentially that's what causes a cell to become specialized. What makes a cell a particular type depends on what proteins it makes, and the proteins that it makes depends on which genes it is expressing, okay? So there's kind of two main ways that we can regulate gene expression in terms of transcription. So this goes back to first semester. Remember, transcription is taking the DNA and turning it into RNA. And typically this is where most of your gene regulation occurs, is at the transcription stage. We learned that back in December when we talked about things like transcription factors. And so there's two main ways that a cell can be kind of told to transcribe these genes. So one is pretty much a fancy way of saying an uneven distribution of chemicals in the cytoplasm. Okay, so asymmetrical distribution means uneven, so like some areas are more concentrated than others. A cytoplasmic factor is essentially a chemical signal. Okay, you could also have differential exposure to an external inducer. So once again, that sounds like a different language. So what we're talking about here is that these are chemicals that are coming from outside the cell. So we could have differences in concentration of certain chemicals inside the cell, or we could have exposure to different chemicals outside of the cell. And that will regulate which genes are getting copied or not. All right, so these are some of the things that can affect whether or not these genes are getting expressed and kind of ultimately determine what those cells will become. As we go throughout the rest of our lesson, we're looking at different things that can affect the development of these embryos, okay? So each one you just kind of need to know from like a vocabulary standpoint. You don't necessarily have to know the specifics. You just need to understand what it is in general and the fact that it can affect gene expression. So this cytoplasmic polarity, okay, this kind of goes back to those uneven distribution of substances in the cytoplasm. But essentially this determines like the basic axes of the body. So like what is up, what is down. And so poles just means a lot of times like a positive or a negative. In this case, your poles are going to have a top and a bottom, okay? So depending on where those cells are they in the top part of that embryo? Are they in the bottom part of that embryo? That'll help them determine what they will become. And so these cytoplasmic determinants, once again, these are just going to be chemical signals that are in the cytoplasm. And early on, when we have this early development, they are distributed unequally. So the fact that there are more of them in certain locations and less of them in other locations, that will affect the pattern of gene expression. All right, induction. So this was the second way that we could regulate transcription. And induction are signals that come from outside the cell, okay? So once again, we have chemical signaling, which is very complex. But this is where we have inductive signals. And we saw induction actually back in gene expression, and it's just chemical signaling from nearby cells, okay? So your signal transduction cascades, this goes back to our cell communication notes, in that you had reception, transduction, response. So essentially you've got chemical signals that are going to bind to receptors on the cell, and that's going to trigger certain responses inside of that cell. All right, pattern formation. Now this has to do with the spatial organization of tissues. And so we kind of have to, the cells kind of have to know where they are, and that in many ways determines what they will become. So like early on in that embryo, when you have the blastula that pinches in, you get these three germ layers, the endoderm, the ectoderm, and the mesoderm. And so depending on where these cells are in different areas of those germ layers, different areas will become different parts. For instance, the ectoderm, the outside, will become the skin and the nervous system. The middle layers become things like muscles and bones and blood, okay? And then your innermost layers become the linings of like your digestive tract. And so just kind of knowing where they are, these spatial differences, that's going to determine kind of what types of cells or what types of systems they are going to become. Another important process is apoptosis. And this was covered back in cell communication, but this is that programmed cell death. So this is kind of like when you have the webs between your fingers that disappear. So certain cells, depending on their location, will become activated to trigger apoptosis and die. So this is actually part of a normal cell development. All right, morphogens. Okay, now morphogens are inducers. So once again, these are chemical signals that come from the outside. So they diffuse from cell to cell. And here we have another concentration gradient. So it has to do with differences in concentration of different chemicals. And so this can help provide some spatial sense to the cells and helping them determine where they are. And then it does play a role in that determination of their fate. So this would be another example of these chemical signals that are coming from outside of the cell. So certain morphogens, they're going to affect specific target cells, and different concentrations will trigger different effects, okay? As you're studying this, once again, kind of study it from a vocabulary type of standpoint. all of these things are going to play a role in cell differentiation and development, okay? All right, so honestly, I don't fully understand this picture, but I just kind of wanted to show you what it's talking about on morphogens here. So here you have the locations of cells. You can see that each one is exposed to different concentrations of these chemicals on the outside, and that will trigger different gene regulation on the inside. That's really all you have to understand about that. All right, Hox genes. We've actually talked about these before. Hox genes are these genes that are shared by animals, and they're essentially body plan or master control genes. If you haven't already, you're going to see these in your What Darwin Never Knew video. And Hox genes essentially regulate other genes. They're kind of like genetic switches. All right, so they kind of tell other cells what to do and what to become, okay? So it's kind of a body plan thing. These are the ones that maybe determine like your basic, where is the head, where are the arms, and mutations in these Hox genes could lead to really some distinct changes in the body plan. So here you have the fruit fly. You can see that this basic color pattern are these Hox genes, and they're going to regulate and turn on other genes within that animal. All right, so when it comes to multicellular organisms, these animals share molecular pathways. So once again, across wide ranges of animals, long spans of time, you are going to see these similar molecular pathways. And this is one of the things that the video that you're watching is going over, is that, like the genes for spots or coloration, things like that, they're very similar. And so certain developmental mechanisms, they've been conserved over long periods of time. They've had just very few and minor changes. And so these are what we kind of consider our basic genetic toolkit. And so just some small modifications or large modifications, some reshuffling over the years is what kind of gives rise to all the diversity that we see today. And so these genetic switches are another thing that you're going to see in the video. And these are going to be some of the things that you would address in the essay portion. You're going to have questions over those. But these molecular switches are essentially kind of on-off switches for genes. So they involve the promoters, those binding sites for RNA polymerase, the transcription factors, those proteins that bind in order to allow RNA polymerase to bind, enhancers, which are going to promote transcription. But essentially these switches are going to kind of signal cascades. So they determine where and when. So where genes will be turned on and when genes will be turned on. So these genetic switches, if there were mutations, as you're going to see in the video, if you haven't already, mutations in the switches can affect the expression of genes, which could potentially lead to some new traits. And so the same switch could have different effects on different genes. Okay, it really just depends on what the species is. All right, modularity. This goes back to one of those main principles, okay, and that each of these molecular pathways, they are independent from one another, okay? So once again, if you had a change to one Hox gene, it wouldn't necessarily affect all of the other pathways that are controlled by the other Hox genes. So each process is kind of on its own. You can have a change in one area, but it wouldn't necessarily affect the whole organism. Another thing that you'll see is heterochrony. You're going to see an example of this in the video, and this is going to be differences in timing. So I think you may or may not have gotten to it yet in the video, but there's a section on finches and their beaks. And so an example of heterochrony would be that the longer that gene was turned on and expressed, then that beak ended up being longer. Here's an example as well. You have your giraffe necks. And so if you were to look at the vertebra structure in that giraffe neck, it's going to be very similar to the vertebra structure that you have in your neck, but yet it's clearly much longer. And so that has to do with how long those genes are going to be expressed in development. And so heterochrony, if you break that down, chron or chrono, like chronological, that refers to time. Hetero means different. So this word refers to differences in timing. And then these modifications, this is another one of those major principles, okay? These modifications are typically going to be modifications of existing genes or existing structures. So very rarely are we just creating something brand new from scratch, okay? Pretty much all of these evolutionary innovations, all of these new traits, all of these changes over time are modifications of things that already exist, okay? And so these regulatory genes that control development For the most part, they're very conserved. They've stayed relatively the same over long courses of time. But anytime you have mutations in these genes or any of these regulatory processes, you can see changes in those organisms over time, and we can see these development of new traits, new structures, and we can see these organisms evolving over time. And that is going to take care of your Evo Devo. I know that's a lot, okay? I know it's kind of tough. So whenever you're studying Evo Devo, one of the things that I want you to do is I want you to focus on the slides that I told you to star. All right, so focus on the four major kind of big ideas behind Evo Devo. I want you to focus on the four processes of development. And in terms of the rest of the notes, just kind of be familiar with what each of those individual things are by definition. So like what is polarity, what is modularity, those types of things. So I will hopefully see you guys soon at some point this week as we wrap up evolution. If you have any questions, just email me, and I hope you guys have a great day.